Thank you. Thank you so much, Minden. And many thanks to the organizers for this wonderful opportunity. I think this is such a rich gathering you have of biotech professionals from, really, I would say, small, big companies, the regulatory side, research side. I think it's really great to be able to have a conversation with so many interested audience members, because as we'll talk about today, for cell and gene therapy, it's really both the science and the regulation that has moved and evolved together. And I think it needs to continue to be a conversation between the scientists and the regulators. So as Minden mentioned, I am the former head of regulatory policy and content management at Spark Therapeutics. And you may be familiar with Spark because they had the first approved AAV gene therapy in the US, Luxturna, which also then went on to get approval in Taiwan, sponsored by Novartis. So if we go to the next slide, please. I just wanted to highlight that everything that I'm talking about in this presentation is from publicly available sources. So I've tried to put on every slide the websites or any papers, publications from which I've taken the guidances from the FDA website or any other websites, as well as any product information. I'll give a few examples towards the end of my talk. And all of that is actually available through the FDA website. And I highly encourage the audience to follow some of those links and to be able to read up on that. If you go to the next slide, please. So what I will cover in our time together today is, I think it's important to specifically talk about the definition of what falls under advanced therapies, and talk a little bit about the various terminology, the names that are used for cell and gene therapy in different parts of the world. I think already even in the introduction, we heard many things. We heard cell and gene therapy, we heard regenerative medicine, we heard advanced therapies. So we'll talk a little bit about what does that mean and why is it important from a regulatory framework to know how the product is defined in any particular region. And then really for the rest of the talk, I'll focus largely on the US regulatory framework. We'll talk about the current status and what has changed, especially in the last 8 to 10 years. There is now a fair amount of guidance that is actually available. And then I'll talk specifically about why that guidance is important when we think about cell and gene therapy development, because I think there are some different things that are needed from small molecules or biologics development. And we can use some examples of those guidances where they specifically help developers to talk about that. And then we'll talk a little bit about special designations, opportunities for meetings that are available, again, in the US, and then just end on a note as we think about global development. So next slide, please. I think we're probably all aware of just how quickly cell and gene therapies has grown as a field recently. The research around this is actually quite old. So it has taken about 30, 40 years, I would say, from some of the initial trials when it was first generation vectors to get to the last 10 years where we suddenly see a lot of key approvals. The graph on the right from FDA shows how the number of INDs, so new drug applications that have come in to the FDA to allow them to start trials in the US, how much that has grown. And you can see that not only is it the number has increased over time, but really there was a period of time that it was almost doubling over 4 to 5 years. So when you think about the regulatory side of that, you can also understand then why FDA would need to also modernize their framework. They would need to issue more guidances because so many companies were coming to them again and again, asking the same questions, and they were giving the same advice over and over again. Of course, when you have so much of that, it becomes easier to actually put it in a guidance so that everybody can read it and every single person doesn't have to go ask the same question. So I think this is a direct impact of the growing field and the growing number of applications coming into FDA. That is one of the reasons why I think we're seeing so much guidance activity in the past 8 to 10 years. And the exciting thing is that I think this market is only projected to continue to grow. While the number of approvals have already increased, they're predicted to be as many as 20 to 30 new approvals in some future years in the US. Next slide, please. And as we talked about approvals, I really liked this picture because I think it shows both all of the current approvals that existed between 2017 and 2024, but also what you can start to see is at the beginning, it was maybe 1 or 2 approvals every year. And this is showing both AAV gene therapy, CAR T therapies, as well as now the gene editing products that were approved in 2023 and 2024. So you can see not only that the number is growing, but also the types of approvals that we're seeing from really just the initial CAR T and one AAV therapy. We can see now different modalities and even ex vivo products. You start to see now many different types of ex vivo products that are starting to be approved here. I think the other thing that's interesting here, and we'll talk about that when we talk about the guidance, is this is also spans many different diseases and indications. So this is not a modality that, was only applicable to oncology or only applicable to rare disease. We see a lot of different applications and indications that are covered by the recent approvals. Next slide. And in keeping with that, just as we saw the number of approvals increasing and also sort of gene editing becoming a new modality that came into the approvals, this is actually changing even more quickly. So if you think about, the viral vectors, for instance, as I mentioned, some of those trials are very, very old. AAV therapy or viral vectors in general, people had started studying them a long time ago. But it took a long time and a lot of technical improvements, both in the design of the vector and in the manufacturing, to actually get to an approved product. What we see now is that cycle time between when a new modality is discovered or being developed to when the clinical trial starts is much shorter. So for some of the new technologies, gene editing as one example, it went very quickly from the new research being published in journals to actually being able to do a clinical trial in humans and then to the first ex vivo gene editing approval. That whole period of time was much shorter than what was the case for some of the viral vectors previously. And I think this trend will continue because if we look now, in addition to the established modalities, the places where we already have approvals, which is lentivirus and AAV, and some more broadly in the retroviral category, we are starting to see now increasingly non-viral forms of delivery that are also coming into the picture. So both what we're delivering and how we're delivering is changing. The physical methods of delivery, before it was just by injection, either IV infusion or like in the case of Luxturna, it was a surgical injection into the eye. We're starting to see now different physical methods that could potentially be used to deliver these therapies. And that goes hand in hand with the fact that some of these are now lipid nanoparticles, exosomes, all different types of therapies that may not need to be delivered in the traditional way. And what all of this allows us to do, which is the graphic at the bottom of this slide here, is it also allows us to maybe look at additional indications that we've not been able to look at before. A lot of the focus initially, if it was, for instance, for CAR Ts, of course, that was for oncology indications. That has changed very recently. You now see autoimmune diseases, a lot of trials for CAR Ts that are going on there, as well as additional applications that are being explored. And then on the viral vector side, most of it used to be rare genetic diseases. So it was a single mutation that was known. And with the viral vector, you could deliver the gene back in. Now we're starting to see more and more trials for chronic diseases or conditions that are not so rare, are a little more prevalent, and it may not necessarily be a single gene mutation. We go to the next slide, please. So coming now to the definition, as I just told you, because this field is changing, one of the things that regulators have had to really think about too is how do they keep up to make sure that the new types of therapies that are being developed and the new modalities would all still fit under this umbrella of advanced therapy, gene therapy, regenerative medicine, whatever the particular regulator has defined it to be. So EMA, I think, has sort of the most basic definition, which is the one that's on this slide, which is really medicines for human use that are based on genes, tissues, or cells. And they break it down broadly into gene therapy, somatic cell therapy, and then tissue engineering. And this broadly covers, I would say, all of cell and gene therapy and regenerative medicine. If we go to the next slide, please. The US FDA gave, I think, a little more detailed definition. So they actually have a web page which says what is gene therapy. And their definition, again, encompasses the same categories that we saw on the EMA website, but I think they gave a few more specific examples. So what you can see in the picture is that they show you both in vivo gene therapy as well as ex vivo. So ex vivo would be where cells are being taken out of the body, being genetically modified, and then the cells are what's returned back to the patient, versus on the right side with the in vivo, you are directly delivering, let's say, a virus or a lipid particle that encapsulates the genetic material into the patient. And in the list below, you can see that this can come in many different forms. So it could be, direct DNA, circular DNA molecules that are delivered as a plasmid. It could be through a viral vector. It could be through a bacterial vector. Genome editing, as we just talked about, being the latest addition to the set of approved products. And then the patient-derived cellular gene therapy products, which is where CAR-Ts would fall as a category. We go to the next slide, please. But as I mentioned, right, when I introduced this section, one of the things we do have to be careful about is that all of the definitions, when we look at regulators around the world, they're not always the same. Even though the underlying technology is going to be the same, I think as a cell and gene therapy developer, we have to be very aware of what categorization our therapy might fall under in any particular region that we go to. And one of the reasons this becomes important is even if we're just thinking about the first clinical trial that we potentially want to do for this therapy, if we actually want to take that forward, there will probably be a larger scale trial that is required. There might be a global trial that is required. And we don't want to be so focused on just understanding one region and then finding that we actually have to go all the way back and do additional preclinical studies just to be able to do a late stage trial in multiple countries or in a different region from where we started. So I think it's very important to know right at the beginning, what are all the possible countries that we might be developing this therapy in, and how might this therapy be regulated if we go to each of those regions. So in this table, I've just given you a few examples, China, EMA, Japan, UK, and US. And you can see that pretty much all of them have different terminology. The UK and EMA are more centered around the advanced therapy medicinal product, which is ATMP. ATMP is also now the terminology that ICH has started to use. So I think we'll see more of the regulators are starting to coalesce around that because of ICH starting to use it. But in, for instance, Japan, it was always known as regenerative medicine. Japan, as you know, is one of the leaders, especially in the cell therapy technology. And then in the US, once they created the regenerative medicine advanced therapy designation, that also became more of the terminology. The other reason why I think it's important to know what classification the product has is based on that, there might actually be some enabling pathways that are offered only to those types of products. So in the EMA, you actually can go through an ATMP classification process that will say, yes, your product is an advanced therapy medicinal product. And then further, they'll classify it on that slide that I showed a few slides ago. They'll further classify it as somatic cell therapy, gene therapy, or tissue engineered product. With the ATMP classification, you can actually have additional meetings available to you, additional pathways. This is the case in several of these regulatory frameworks. And then there could also be, for instance, with Japan and the Sakigake designation, it's not specific just to regenerative medicine, but it's possible that more regenerative medicine products might be able to go through that pathway, especially if they are for genetic diseases and they know that they have a local population in Japan with that mutation or who would be eligible for that trial. So I would say the biggest takeaway from this slide really should be make sure you don't wait to learn what your product would actually be considered in other regions of the world. I think it's something you should map out at the very beginning, even when you're starting the first couple of trials. We go to the next slide, please. So I'll talk a little bit now about the US framework specifically for cell and gene therapies. And you'll see through these slides how much this has changed, I would say, within the last 8 to 10 years. Next slide, please. So starting at the very top, if you think about what the legal framework is, and I'm giving a side-by-side comparison here between the European Union and the US. And as I mentioned with the EU, it's really has focused around this definition of advanced therapy medicinal product. And there are specific directives, as listed on this slide, that define the actual legal framework. And then within that, they would have a various sets of guidelines for developing these products. In the US, it's really two main acts that cover this, the Public Health Security Act and the Food, Drug, and Cosmetics Act. The specific section in the Code of Federal Regulations, which is the regulation of biologics, that also covers cell and gene therapy. Next slide, please. I think probably everyone in the audience knows this already, but just since we are talking a lot about guidances in the rest of this presentation, I thought it would be good to just remind everybody what we just looked at was the legal framework under which cell and gene therapies are regulated. In every regulatory authority, there is at the very top a law that essentially defines the regulation of those products. Based on that law, the health authority then has the ability to make rules and regulations. So in the previous slide, I talked about the Code of Federal Regulations and the specific section that applies to biologics. That's that second part of the triangle. Important thing to remember is that both law and rules and regulation are actually binding. So you do have to follow what is written and what is defined there. Now, most of the times, as I think any of you who may have read these that are familiar, most of the time these are written very broadly so that it allows everybody to still be able to follow what's written in the law and actually go into more detailed ways of how to go about developing that product in the last two things that are shown on this triangle in through guidances and then policies and procedures. And the advantage with that is that guidances and policies are actually not binding as they would be by law. It's more of a helpful way to help sponsors to understand the health authority's position, help them to share knowledge back with the sponsors. And really, as the word says, it's really meant to guide you, but the sponsor still has the ability to put forth their own position to the regulators and hopefully get some feedback on that. So all of the guidances that we'll talk about in the rest of this presentation, think of it as FDA communicating their latest thinking on development of these products to sponsors. Obviously, you know that is ideally what they want to see. But if there are places where maybe you need to do something a little bit differently based on your product, or maybe it's such a brand new type of product that it isn't even covered in the guidance yet, then you can do your best to try to follow the spirit of anything that's closest to your product and then still go talk to the health authorities to have a discussion about specifically what you want to develop. And then finally, the procedures and policies is more what FDA would use internally. So just as one example, there is a SOPP on specifically the Interact meeting, which I'll talk about in a few slides, and how that should be conducted between FDA and sponsors. The policy is actually written for use by FDA staff, but it is publicly available so that it gives sponsors an understanding of how will that meeting take place, and how should you go about preparing for it. So next slide, please. The FDA specifically in the US is organized into centers, and then within the centers, offices by area of expertise. As many of you know, there have been a lot of changes with the US FDA, especially in this current year. So I tried to give you as updated an idea as possible. I think this chart is from September. And it shows all of the new FDA leadership. So everyone from the commissioner to each of the center directors has actually changed recently. That's shown on the slide. And then really the three main divisions that any of the therapeutics would fall under would be CDER, CBER, and CDRH. So CDER, the Center for Drug Evaluation and Research, has essentially all of the small molecule drugs, and then therapeutic biology. So monoclonal antibodies that are now well understood and developed are also under CDER. CBER, which is the Center for Biologics Evaluation and Research, that's where all of the cell and gene therapy, vaccines, and blood products are regulated. So if you are developing a cell and gene therapy, it will be reviewed by CBER. And then finally, the Center for Device and Radiological Health, that's all of the medical devices, diagnostics, and radiology products. I think it's still very important to pay attention to the things that are regulated by CDRH, even for gene therapies, because a lot of times it is possible that you may need a specialized device to deliver a gene therapy. For instance, for neuroscience applications, if you need delivery into the brain, there could be a specialized device that's needed. There could be a surgical procedure that's needed depending on the disease that you're targeting. And there could also be diagnostics that need to be developed together with the gene therapy, either for screening patients or for making treatment decisions, or as one example for viral vectors, to understand if there are any pre-existing antibodies against those vectors that would, again, allow patients to either be included or not included. If any of these diagnostics tests are a decision-making part of developing that therapeutic, if they're used in the diagnosis or treatment in any way, then those will fall under CDRH and will actually be regulated as diagnostics. This is an area that is very confusing. It has been changing a lot, even on the US side, to understand what's actually been regulated or not. So I would highly recommend that if you are a sponsor of a gene therapy and you're considering doing the trial in the US, please talk to someone who is knowledgeable about the diagnostics framework. Do it very early, because it could possibly be that you actually need to develop your companion diagnostic or co-diagnostic together with your product, and that you may not actually be able to start your trial in time if you don't already have that developed. There are people whose entire job it is just to keep up with what's happening with the diagnostics framework in the US, and I highly recommend having a conversation with them when you think about how to develop your gene therapy product in the US. So if we go to the next slide, please. So focusing more specifically on CBER, which regulates cell and gene therapies, within CBER, they actually created what they call the Office of Therapeutic Products, or OTP. If you've seen any of the US regulatory framework talks recently, you might have seen this abbreviation OTP. That was created in 2023. The important thing to remember is it was created under the previous administration and the previous FDA leadership, and a lot has changed since then. So while, as far as we know, the Office of Therapeutic Products is still there and they're still regulating gene therapies, all of the top leadership has changed. The person who was leading OTP, as well as her deputy, were put on administrative leave earlier this summer. They brought in new people to lead OTP. So I think, again, there are some question marks now around, what's the philosophy? Is it the same type of openness that the previous OTP leadership had? I think what we're hearing from FDA is still, yes, they're very interested in cell and gene therapy products. They're very interested in partnering with sponsors. So hopefully all of those things that were started when they created OTP, it's really our hope as sponsors that it continues to be that collaborative environment going forward. But again, I would say if you are a sponsor that is planning to have a meeting with the US FDA in the near future on a gene therapy product, please pay attention to what's going on with the changes of FDA and keep track, because it's very dynamic. It keeps changing even now from, a week to week basis. The great thing about OTP being created and the fact that it still exists is that it's what's called a super office. And that basically means that it would allow for increased number of applications to go in. If you think about where I started the talk with the graph from FDA on how the number of INDs have, doubled and tripled. Obviously, they need the review capacity to be able to do that. And then this also, having a super office, allows them to have multiple disciplines and to be able to get that alignment across those disciplines and cell and gene therapy experts, no matter where they sit in FDA. So you could have cell and gene therapy experts that were in ophthalmology or oncology or hematology. When you think about, some of the recent indications, this is another way to, again, bring that expertise from all those divisions together and to be able to, for the internal reviewers to also be able to learn from each other. The OTP structure is also then further divided, where you can see that there is, really think about this as sort of the disciplines. So you have, the folks who are looking at the preclinical data. You have the folks who are looking at manufacturing data. Toxicology, obviously a large focus of the clinical data. And then you have the clinical review divisions based on the indication or the disease that's looking at that. And then, of course, you need to have the project managers and the administrators to actually interact with the sponsors on all of these things. I would say continue to monitor this, because this structure is something that definitely could change and might change in the future, depending on how FDA as a whole continues with either the current structuring or whether they have another reorganization. So we go to the next slide, please. So I know this is a lot of guidances that I've listed here on this slide. I don't expect you to really read through this. I really want to make just two important points when you look at this. The first is I've divided the slide up to show the large period of time, essentially between the late 1990s to 2019, and there were 20 guidances that were issued that had anything to do with cell or gene therapy or regenerative medicine. And there are some representative examples there, preclinical, there were some disease-specific guidances, but there was not a lot that people could really follow. And then on the right side, what you see is just between 2020 and September 2025, so barely 5 short years, you already have over 15 new guidances that are specific to cell and gene therapy and regenerative medicine. So the point I want to make here is this is evolving rapidly. The regulators are gaining much more experience because they have so many different products that are coming in to start clinical trials that they're seeing, and they're able to translate that experience looking across so many different applications to saying, what's general guidance that we want to be able to give sponsors? And the more guidance that there is out there, the more helpful it is for sponsors, because, again, it's going to give you a starting point rather than having to go to FDA to ask every single question. And then it's also going to be helpful for the FDA reviewers, because hopefully they're not answering that same question for every single sponsor, because they've already read the guidance and they have some idea how to go about it. So I think the key takeaway here is that FDA has really been responsive to the number of new applications and responded to it with guidance. And I think the second takeaway that I would ask you all to remember is that probably means that there is at least some level of guidance out there for your product or your indication. If you have a completely novel modality, it may not exactly speak to what you're doing, but I would say make sure that you're familiar with this list of guidances. Make sure you visit the FDA webpage where these guidances are listed, and try to read as many that are as close to what you are developing, because it will be important to show that you have understood FDA's viewpoint when you go in to have the meeting. When you're asking to do something different, especially from what they're asking you to do, it's important to have demonstrated that you know what the FDA viewpoint is, and then make your argument for why you would want to do something slightly different or additional to what they're asking. So if we go to the next slide, I'm going to give you examples, 3 specific examples now of what has changed when it comes to issuing this guidance. So one of the things that has evolved is specific therapeutic areas became very popular, especially for the initial cell and gene therapies that were being developed. As I mentioned, there were some certain areas where there were either single gene mutations that were known, or it was very clear what was missing and could actually be replaced with the gene therapy. And for 4 of these that you see that are very disease-specific guidances that exist now: gene therapy for retinal disorders, for hemophilia, for rare diseases, and for neurodegenerative diseases. So if what you're developing for falls into any of these categories, you absolutely need to make sure that you are familiar. I would also say the human gene therapy for rare diseases is just a good guidance to know, because it's broad enough, since it tries to cover a whole umbrella of rare diseases. It's broad enough that I think you can get some general guidance out of it, even if what you're developing is not in any of these therapeutic areas. Next slide, please. The second example is how guidance has had to evolve with how the science has changed and how much we have really understood now with, as I mentioned at the beginning, 20, 30 years more of doing gene therapy clinical trials and really taking all of those learnings. And one of the examples that we've seen here is the long-term follow-up guidance. So gene therapies, and cell therapies, because of the fact that they're really a living therapy that you're delivering into the body, it's actually going to stick around in the body for a really long time. So you think about, if we take tablets, if we take aspirin, you have to take the tablet very frequently, every so many hours, because it's essentially being metabolized in your body, and the therapeutic effect only lasts for a few hours. For certain biologics, they might have a much longer half-life within the body. So for instance, maybe it is a monthly infusion or injection, but still after that month, you're going to have to go get another injection or infusion. Otherwise the therapeutic effect would drop. Cell and gene therapies can look very different. They can be very, very long-lived. And part of the science that we're still learning is exactly how long, right? Exactly how long might those cells stick around? Exactly how long might the vector continue to express the gene at a level that it's actually making therapeutic levels of protein. So this guidance framework, which I think is very important for everybody to be aware of, because I think no matter where in the world you're developing your therapy, I find this checklist pretty useful, is you're going to think about your product, how it's delivered, what's actually going into the body, whether it's actually integrated into the genome or not, how long you expect it to persist. And then based on that, you're basically developing essentially a risk categorization for your product. And based on that risk categorization, that will determine how long after your clinical trial is finished, you need to continue to follow those patients, because there could be certain safety events that maybe don't emerge within the duration of the trial, whether it's a few months to a year to a few years. It's still not long enough to see what long-term persistence of that vector might be doing. I really have to wait a lot longer to be able to see that. And I think this guidance is a great example of how would you determine that risk categorization and how long you would need to do the trial. This is something else I think sponsors really need to be aware of before you start the trial, because you are committed to the long-term follow-up. It is not easy for a cell and gene therapy to say, okay, my trial's done now. I have my 18 to 24 months of data, or how much ever data that you actually need. Now I can just close out the trial like I would do for other therapies. That's not the case for a lot of cell and gene therapies. You are following these patients sometimes for 10 to 15 years. So it's important, even for a phase 1 trial, to know that that might be the case and to actually make a plan to be able to do that. So next slide, please. And then the last example I have here, and this is a complicated concept. I probably am not going to be able to spend as much time on it, this is another place where if you are developing something that you know is using a similar vector to something that's already out there, especially similar to an already approved drug, I would highly recommend that you talk to experts who are familiar with the orphan drug regulations and are familiar with this guidance. But this is another example where FDA really had to put this guidance out specific to the gene therapy modality, because if you think of other types of products, if you think of small molecules, it's very obvious based on the molecular structure whether those two molecules are the same or not, right? If you change a particular specific molecule within that, does that create a brand new compound, or are you making another version of the same compound? FDA had also done some work actually to define this then for antibodies based on essentially the sequence of the antibody. So again, you would know whether changing the sequence of changing something in the sequence of an antibody was actually a consequential enough change to say, hey, this is a whole new product now, versus, okay, this is a minor change and it's essentially still the same antibody as it was before. But none of those rules for small molecules or antibodies could really be applied when we think about gene therapy. So FDA had to really put a whole new framework in place to say, when would something be considered the same drug versus a different drug when we're talking about, for instance, a virally delivered gene therapy. And what you see on this slide are some specific examples or some specific factors that they've called out to say it would be a different drug if you change the transgenes, if you change the vector group entirely, and then some guidance on how would you define it as a different drug, even if you were within the same vector group. And then on the other side, they're telling you what would not be considered different drugs, or if they were essentially just minor differences, or if it was the same vector and same transgene, and the changes that had been made were much more superficial. So again, I'm not spending nearly enough time on this for what is a pretty detailed topic, but I think it's something else that sponsors should be aware of depending on what you're developing and how similar it might be to existing technology that's already out there. Okay, let's keep moving. If we go to the next slide, please. I just wanted to quickly mention, I'm not going to spend time on this on the next slide, but just wanted to give you all a visual to say that in addition to all of the guidance that's available from FDA, there are specifically flowcharts and checklists specific to advanced therapy medicinal products, which, as we said, is where cell and gene therapies falls for EMA. That's available on the EMA website. And I think they're very useful because they are both in the form of a flowchart and then actually gives you a checklist that you can check off to say, based on the stage of development that you're in, am I actually able to have data around all of these different topics? So if we go to the next slide, please, you'll see an example. So this is an example of a flowchart around non-clinical development. It asks some key questions about what is being developed and what is the intended use in the patient. It then tells the sponsor what you actually should be doing if you have that type of product, and then further lists the guidances as well as regulations that are applicable to those. So I think it's a great way to see everything in one place. And I think you should definitely take the time to look at each of the different flowcharts that are available. And then if we go to the next slide as an example of the checklist. So we go to the next slide, please. Yep, perfect. So this is now on the manufacturing side. This is a quality checklist, which tells you these are all of the things that you actually need to be doing. So make sure you're thinking about this early. The potency assay, which is the very top item, is a really important factor that comes up because the mechanism of action for a lot of gene therapies is much more complicated, right? It's not always easy enough as saying, I'm just going to biochemically measure one enzyme or one factor. And if so much of that is produced to a certain quantity, then I know that my product is potent. A lot of the times, by re-expressing a gene, you're turning on an entire biological pathway. Many things are happening in the cells. Either, you're reintroducing the gene, or sometimes you might be silencing something. You might take something away. That leads to many, many biological effects in the body. And how can we really create a measurable, repeatable assay that would allow the potency of that product to be studied? is something that I think sponsors need to, again, start working on very early and not wait until they're going to go into late stage trials to be able to think about that. Okay, let's go to the next slide. So in coming to the second half of the talk here, I'm going to talk through some specific considerations when we're talking about cell and gene therapy development. And what I'll do here is going to talk through each of the different stages of development, what the considerations are, and then we'll look specifically at where there is guidance available to be able to help drive how you would work on some of those questions that come up. So go to the next slide, please. I think we've kind of touched on a lot of these as we've talked about already, but one of the things that's important to remember is that cell and gene therapies are not like other products, like small molecules or antibodies. We heard at the very beginning of the seminar how much potential these actually have to meet unmet medical need. You're now being able to tackle diseases and indications that otherwise had absolutely no hope in terms of medicine. And now there might actually be something available, even if it's a very rare disease or a very small population. So there is a lot of hope that is created by cell and gene therapies coming into the picture. These can also be potentially a one-time administration. We talked about how the effects could last a really, really long time. That could mean that you are basically getting it once in your life, and then you don't have to ever have another injection, infusion, surgery ever again after that. The clinical effects, especially if you are able to really restore a biological pathway that was misregulated based on these, the clinical effects seen can be really huge. And even in the earliest of trials, a lot of times even in phase 1 trials, which for cell and gene therapies very often will end up being in the disease populations, because if you're targeting a mutation, obviously you need someone to have that mutation. Or if, let's say, for a CAR T, if it's for an oncology indication, of course, you're going to go into patients. So a lot of the time, even in phase 1 trials, you actually know this is working, and it's working really well. And that effect size can be seen as being pretty large, even with a small number of patients. And then finally, we know the regulatory authorities are also equally excited, right? If those top 3 points that I talked about are true, if you're going after something with a huge medical need, if it's something that can eliminate the clinical burden of treatment from the rest of the lives of the patients, otherwise they would need hours and months and multiple visits to the hospital, and now you're talking about a one-time treatment. And if we know clearly the data shows and the statistics show that it's working, then obviously regulators also get excited about these things. And we know that they're committed to partnering with sponsors to develop these therapies. So that's all really exciting, the promise of cell and gene therapies. But then what are the considerations when you start to actually develop them? We talked about the fact that they're durable and can last a long time. The thing to remember if they last a long time is that with currently all of the approved therapies that are on the market right now, there is no way to turn it off. Once you've actually delivered the therapy, it's now being delivered. There is no off switch. There's no ability to say, okay, maybe this is not doing for this patient what we thought it was going to do. So let's just, silence the vector, just no longer expressing the gene, or let's just take all the CAR T cells back out that we gave. Currently, there's no way to do that. So once it's been delivered, we have to be able to monitor and know what's going on in the body. And that's really the second point, which there's then a necessity for long-term follow-up. And then depending on how that vector or that therapeutic is distributed into the body, you may potentially need to track not just the patient, but also the patient's partner and any future pregnancies that may arise after delivering that therapy. The trials themselves, while the clinical effects might be large, the trials can be a lot trickier and harder to do because you might have a really small number of patients. In order to have that really clinically meaningful data that's also statistically significant, you may need to have very clear predefined endpoints. And if the disease is rare and the patient population is small, those endpoints may not exist. A lot of times you're studying the disease as you're developing the therapeutic. You're understanding what happens to the disease as you're developing the therapeutic, which obviously makes this trial design a lot harder. Manufacturing, I think you could do a whole seminar that's dedicated just to how complex the manufacturing of these therapies are. And it's something that really sponsors need to partner very closely with regulators to get their input and their interaction to make sure that the manufacturing process they're developing early is actually going to be the same one that you can use later on. And then non-clinical studies, which we'll talk more in the next slide about this. There's a lot of traditional methods that are used, traditional toxicology, traditional PK/PD, which is simply not applicable here. And then the last part I would say is as much as regulators are excited and evolving and want to work with sponsors, depending on where you go and what type of modality you're developing, it could also mean that the experience with reviewing these applications could be limited. So it might take more conversation, it might take more back and forth to make sure that both regulators and sponsors are on the same page. But I have to say, even year to year, this is changing rapidly. So I think increasingly now regulators around the world do have experience with cell and gene therapies and are able to give meaningful feedback even when it comes to new modalities. So next slide, please. Let's now talk through just the various stages of development and what some of the considerations and guidances are. There's going to be a lot of text on the slides that are coming up. I'm just going to call out what's important and then spend a little more time on the guidances. But if you really think about traditional discovery to commercialization, I think we've all probably familiar with these phases, starting from discovery to preclinical to the various stages, phase 1, phase 2, phase 3 of clinical development, and then approval and being on the market. This can all blend together pretty significantly, especially the phases of clinical development for cell and gene therapies. And you'll see those considerations in the next few slides. So if we go to the next slide, please. So starting at the very beginning, when it comes to discovery, and we talked about this at the beginning of this talk, how much this is changing and how important it will be to know whether what you're using in terms of the vector, the serotype, the cell type, the transgene, how the construct is actually put together, whether this is something that's completely new or whether there is actually existing data from any of the approved products or products in development. If you are developing something that's truly new and nobody has ever done it and nobody has any clinical data to support it, the level of preclinical evidence that you're going to need to have is going to be higher because there is no knowledge of the safety in a human population. If what you're developing is very similar to something that's already an approved product or where there are many clinical trials ongoing, you will still need to generate the data for your own therapeutic to be able to file it. But at least you'll have the added advantage that there are, potentially publications or potentially a larger body of knowledge that's out there. This is also a really good time at that discovery stage to understand if you were to actually go forward with making it, especially if it's a brand new vector, will you actually have the manufacturing system, the ability to deliver it? Will you actually know that it's going to the target tissue that you actually want it to go to? These are all of the things you want to actually think about very early when you're designing the construct, because regulators are going to come back and ask you those questions. The more new what you're doing is, I think the more exciting it could potentially be, the more data you're going to need to convince regulators for why that new modality should actually go into trials. Next slide, please. So as you think about, now let's say you've designed that construct, you are feeling confident about whatever the new elements are, maybe some of the design is similar to some things, but then you've added some new elements into that. Then you're really going to think about what is that preclinical package going to look like. As I mentioned on the previous slide, traditional PK/PD or tox really may not work here. Instead, there are very clear guidance around what you do need to be able to do around biodistribution, tumorigenicity, the shedding of the vector, as well as finding out whether it's actually going into germline tissues or not. Because if it is, then as I mentioned before, now you're actually tracking not just the generation of patient that receives the therapy, but you may need to track the next generation as well. So if you want to do all of these studies, the choice of your non-clinical species becomes really important. You may not, there's no classic toxicology pathway here. of doing, a small animal or a large animal. Mouse or rat studies may not make sense based on what you're doing. Your target may not be expressed in that clinical species at all. So you really have to think hard about what is the data what is this study actually going to tell me? And is it actually going to help me answer the question of whether this is safe to move into humans or not? So if we go to the next slide, you'll see that a lot of this is actually specifically covered. So some of these are directly slides that FDA leaders have presented previously. This is one of them. And what I've done at the bottom is where you see that little folder icon, I've added some of the guidances that you may want to look at as a developer that would definitely cover these topics. So, of course, the guidance for industry on preclinical assessment. This very specifically focuses on all of the considerations for preclinical development. But in addition, you probably want to look at if you're developing any of those specific diseases, whether it's a disease-specific guidance, you definitely want to take a look at those as well. And a lot of this will cover some of the things I just talked about on the slide before. How do you look at the animal model? Should it be a healthy model or a disease model? How do you look at where the cell or the vector is actually going and how it's being distributed into the body? How can you mimic the route of administration to be as close to what it's going to be in the clinic? And then, of course, how much of the normal toxicology experiments with the profiles would you need to do? Where else can you look for guidance? And again, this is a place where I think the feedback from FDA becomes really important. Okay, so we go to the next slide. I'm going to speed up a little bit here because I think otherwise I'm going to run out of time to finish everything. But essentially what this next set of slides in this section does is it goes through every part from that early clinical development to late clinical development and asks the same set of questions. What's actually unique about cell and gene therapies? And where are there guidances that we might be able to look at this? So for a first in human study, what's the design? What type of patients are we going for? How do we choose the dose and the endpoints? If we go to the next slide, please. This is captured specifically in the long-term follow-up after administration of human gene therapy products guidance. Both of the topics that I just talked about, really focusing on what's that initial dosing, how do we find that, and then what are the safety considerations? Because remember, with gene therapy, most of the time, almost all of the time, you are directly going to go into patients. So this is not just a tolerability study or a healthy volunteer study. You're almost always going to be directly looking at what happens to the safety profile in the patients that actually have the mutation or the condition that we're going after. And then for all of these, again, even if it's an early phase trial, you will need to do the long-term follow-up and the tracking. If we go to the next slide. So that's some of the same questions then move through late development, but you're adding now additional questions around really having a large enough trial, even if it's a very small population, to be able to show the effect size that you would need. And then let's go to the next slide. And that will move you through some of the questions around post-approval and commercialization. What is the risk, that long-term risk that we've been talking about? What are those post-marketing requirements and commitments? What kind of long-term follow-up would you need? Are there actually registries that would need to be developed or additional phase 4 trials? And then from a manufacturing side, how do I keep making the drug in the same way that I was making all along? I just want to make sure you can still hear me because I just lost my earbuds. Yes. Here. All right, so let's go to the next slide. And all of that is captured specifically in the modality and disease-specific guidances, as well as the long-term follow-up guidance. There's also relatively new guidance specifically around post-approval methods to capture safety and efficacy of cell and gene therapy products. And a lot of this has to do with, even if you give this therapy once, it actually needs to have effectiveness that can actually last to really be able to benefit the patients for that specific disease. It is all going to differ here between rare and common diseases, as we talked about. For rare diseases, maybe a single-arm study is enough. You will probably see a large effect size, and that will still give you the statistics that you need. But if you have more common diseases and much larger populations, then you may need to do much larger size trials to be able to see that same effect. And then I've already talked about companion diagnostics and devices. So next slide, please. So this is just the slide which shows all of the considerations that we just walked through in one picture. So if you ever want to just be able to show, in one slide, a snapshot why it's complicated to develop cell and gene therapies, this is usually the place that I go. And then it also has some additional manufacturing considerations. So if we go to the next slide, you'll see the guidances that specifically call out CMC development. And this is based on both manufacturing and product characterization. So what all do you need to think about when you're actually making the product? How will you actually characterize the product that you've made? And then another really big consideration here is manufacturing changes, because you may want to make changes based on what you have learned, but you have to be very careful as to how those changes might actually change something fundamentally important about your product, and what impact that might then have on the clinical activity of that product. Okay, so if we go to the next slide, and I know I have a couple sections here, so Xiaoying, I'm going to try to go through it quickly to make sure we still have time for questions, but I'll just highlight some of the things in this section. So if we go to the next slide, what I've called out here, and actually I'll just focus on this slide to summarize, is specifically with the FDA, there are a lot of opportunities to actually get feedback and to be able to engage on a cell and gene therapy product. You can see the various types of meetings laid out here across that development timeline. A lot of them are, standard meetings that you would get for any type of product that you might be developing. It doesn't have to be specific. But some of the things that are specific to cell and gene therapy products is the CAT meeting and the Interact meetings. If we go to the next slide, the CAT meeting especially is something that happens very, very early. You can just essentially have a product concept. Can we go to the next slide, please? Oh, sorry, one more. There we go, perfect. This is something that is very early. It's a 2-page meeting request, which is why I want to highlight it, because you could very much be at the conception stage of as you're thinking about what your innovative product might look like, and still be able to get some very early feedback on your novel technology. So if you're truly new to this field and you're just starting out with this and you think you have a novel technology, this is definitely a meeting type that I would recommend. The Interact meeting, which I skipped over in the interest of time, is also another important one. It's not specific to cell and gene therapies now. It is available to everybody, but you can actually do that type of meeting before you design your pivotal toxicology studies. And FDA will actually give you feedback and tell you what it is that they want to see in order to be able to move the product into clinical trials. So let's go to the next slide, please. In addition to the Interact and the CAT meetings, there's also a meeting type, relatively new meeting type called Type D meeting, where you can ask very specific questions. So unlike some of your end of phase meetings where you have lots of different disciplines involved, the Type D meeting is really just for one or two disciplines, like a CMC question and a preclinical question, or let's say a clinical and an CMC question. It will allow you to have more time to spend on those focused questions. And these will be granted because there's a timeline around it that's required by PDUFA. There's also a mechanism now to ask for requests for clarification. So if you did get feedback in one of your normal meetings, but something wasn't clear, or you felt like you didn't really agree with FDA, you can take just that topic and send in a request for clarification, and hopefully avoid a sense of where, maybe you thought you heard something, but the next time you go to FDA, they say, oh, no, that's not what we agreed to. The request for clarification can really help you do that. Okay, we go to the next slide, please. I'm just going to mention quickly here a pilot program that FDA started. It's called the START pilot. Again, it's not specific to cell and gene therapies, but has been very important, I would say, for some of the cell and gene therapy programs that have gone in. You can see on the slide the criteria that are being used, but the advantage of this is that it allows for a lot of frequent communication back and forth between sponsors and FDA, much more than you would be able to get even with, the slide that I just showed with all of the meetings. Those can come sometimes years apart in your development program versus the START pilot, which is really meant to have continuous back and forth. You could have a assigned, or you always have an assigned project manager, but you're basically going to have a dedicated person who is always checking for that communication and turning it around to you quickly. So I think this is an important tool to keep in mind if you do qualify for the criteria. Let's go to the next slide. Let's skip this one because this is focused on EU, but just be aware that EU has its set of meetings as well. And then what I'll just highlight here, and I mentioned this at the beginning when we talked about the definition of the therapies, is there can be several enabling pathways and designations that are available to you, either to any type of therapy in that region or specifically if you have a cell and gene therapy. So examples in the US would be the Regenerative Medicine Advanced Therapy designation. That is specific to if you have a cell and gene therapy. It will give you essentially all of the benefits of the other designations that are listed on here. And then, equivalent in the EU and the UK, a lot of these sometimes focus not just on regulatory feedback, but also payer feedback to go along with this as well. And if you go to the next slide, please. This is, I won't go through all of the details here, but what I wanted to do was give you all a sense of, if we look at different types of recent approved products when it comes to cell and gene therapies, and I've intentionally picked here an AAV-based gene therapy, a gene therapy that has accelerated approval instead of traditional approval, gene editing product, and then a CAR T therapy product. And I would encourage everybody, when you get the slides later, to really take a look at this, because what you'll see is all of the different paths that these sponsors took to have their key interactions with FDA. When they did their key meetings, how much time it took from the time they started filing their BLA to actually get the data, to have the important mid and late cycle meetings, and then actually leading to approval. And the biggest takeaway from all of this is that you'll see that no two of them are the same. No two of them, even though they're all cell and gene therapies, no two of them applied for exactly the same designations or went through the exact same timeline. And I think it tells us two things. I think it tells us there's no one universal formula for doing this. It really does depend on your type of therapy, your indication when you think about how long that trial is going to take, when you're actually going to have the data. And then some of this, as you'll see with the Luxturna example, is it used to take much, much longer. 8-10 years ago, you think about how long it took from the initial pre-IND meeting to get to approval. It's over 10 years. That's not the case anymore. Think about our gene editing product example that's here. This can all start to happen very quickly now. You can go down to 5-6 years of development with some of these therapies. And you can see if you look through, and all of this, by the way, is publicly available on the FDA website. You can look at every approved product. You can go through the approval documents and understand all of the mechanisms and the conversations that they've had. And you'll start to see that this is starting to get much, much faster. Let's go to the next slide. Shaling, I think in the interest of time, maybe I'll just summarize this section to say, I know the focus of the seminar was really meant to be on the US anyways. So what I really had in these last 3 slides was to remind everybody that it's not just the US, it's really the global regulatory framework that's also evolving. We have many regions now that didn't have guidances for cell and gene therapies that actually now have guidances. Where there used to be no approved products, I think we're starting to see some products that are approved in 40, 50 countries around the world, even though they're cell and gene therapies. So I would say just keep that in mind as you're developing your therapy. Think not only of the first region that you're going to go to, but think about all of the regions that you could potentially have an impact in the world and how you might design your trial accordingly. based on that. So let's just maybe, can we skip through the next 3 slides? Just go to the summary, please. Perfect. Thank you. So maybe that's where I'll stop to say, I think the biggest thing here is, it's really knowing 3 things about what you're doing. It's knowing what your product is, whether it's an existing modality or a new modality. And even if it is a new technology, I would say, don't feel like that's necessarily going to be a barrier from a regulatory perspective. I think that could actually be an advantage in the current regulatory environment and have regulators very interested in talking with you. But make sure you really understand it, understand what your primary mechanism of action is, because that will determine your regulatory classification. As we've talked about, know your development plan. The initial country where you start the clinical trials is just the beginning. Think about the whole picture and think about it early, and what other regions you might need to know about, because their regulatory framework could be evolving very rapidly. Think about all of the opportunities you have to get health authority feedback, either in that first region or by having joint meetings where possible, because the more you know about how regulators are thinking upfront, the better plan that you're going to be able to have to be able to do that. And especially this time on high unmet medical need or rare diseases, think about all of the mechanisms that could help you do this faster. And then I'll finally stop to say, at the end of the day, I know we're all doing this for the benefit of patients. So even when it is hard, even when it is a complicated regulatory pathway, just know that it's worth it, because these really are life-changing products for a lot of people.